All right, well, thank you everybody for coming. I want to introduce our seminar speaker, Nate Grubaugh, today. He's come from Yale, and Dr. Shu has collaborated with him, sent samples to him, sent samples to his lab, to Chantel Vogels, who's now independent from your lab and is a assistant professor. We've had this interaction with the lab for a while. He's coming from Yale, so it just made sense that he'd come up. And we have a bit of a relationship in that Greg Ebo, with whom he did his PhD at Colorado State, I knew in a lab. We worked at a lab, not at the same time. We worked at a lab at Harvard together. So I thought it was a connection. I thought, well, but then I realized that you started in the lab after Andy Spielman. As a frame of reference, the PI of the lab we worked from died in 2005. And Nate said that he was just graduating undergrad at that point. So we're a whole generation apart, even though we have that connection. So time just sort of creeps up on you. So before he was at Colorado State, he did a master's at Hopkins and then did an undergraduate degree at Western Michigan. Mm-hmm. And he then went to Wadsworth Center, worked with Scripps, sorry. I'm getting you mixed up with
Alex Yoda.
Yeah. And now he's at Yale, where he's an associate professor, and he's going to tell us about some very interesting work he does with viruses and mosquitoes. So thanks. Welcome.
Thank you, Steve, for inviting me. I really like to have these, connections within our region on a lot of the work that here. Thank you, Nolan, for picking me up this morning and showing me around. And thank you all for being here. Excited to meet some of you later on today. So I'll give you a quick intro on my lab first. So some of the main things that we do is that we sequence viruses and then use phylogenetic tools to understand how they emerge and spread and cause outbreaks, cause disease. My true passion is what you see up here is mosquito-borne viruses, but we have expanded that in a lot of ways. I study a lot of respiratory viruses now. SARS-CoV-2 out of necessity. In fact, we sequenced 25,000 SARS-CoV-2 infections during the pandemic, set up a high throughput sequencing lab. I now even do work with influenza, RSV, and human metapneumovirus. Really just interested in these patterns of importation and spread. And then my bigger international program is focusing on dengue virus. I have a NIH New Innovator Award to build genomic tools to create a global dengue genomic surveillance program. I have a lot of field sites in places like Colombia and Puerto Rico. I work with the Florida Health Department on this. And I really like to understand how do we use genomics to help inform public health. But really my true desire, my true passion is what I'm going to talk about today. It's more about how do we use genomics to understand ecology, how viruses are maintained, how they persist within our environment. Okay, so some frame setting first. This is a phylogenetic tree. Y'all probably know that. The key thing that I want to make sure that y'all know going into this talk is what I'm going to be talking about throughout is this concept called phylogeography. So when we construct a phylogenetic tree, you have two sequences or a cluster of sequences, and based on their genetic relatedness, shows the branching pattern in the phylogenetic tree. This one is a little bit special in that first, it's time resolved. So every sample that we have sequenced is on this x-axis by its time that it was sequenced. And then this here would be called an internal node. Where they come together is then based on, this is a estimated ancestor that existed in time. And where that is placed is based on its evolutionary rate. So we know how genetically distant these things are. We know how fast they evolve over time. So we can place their ancestor back in time. Right? So first we're creating time-resolved phylogenetic trees. The next thing that we're doing in here is, this is called discrete trait analysis, and the trait is location. We're trying to estimate where these ancestors occurred back in time. So all of these sequences are colored by the state. By the way, this is West Nile virus. I'm sure most of you looked at that and already assumed that was West Nile virus, but no. So this is West Nile virus from throughout the United States. They're colored by state. And so when all of these here are Louisiana, the ancestor node is like, well, that's pretty easy. That's probably in Louisiana. It gets a little bit more complicated when you have all of these states in here. So in this tree, based on the genetic divergence of the sequences and where they're located and when, we estimate that this all of these sequences originated in New York and had spread throughout there. So then when you have time and location, you can also plot these on a map. And if you go through a tree, you can actually then recreate how West Nile virus emerged. It was first introduced into the United States in late 1998, finally detected in 1999, and how within a few years it had spread across the United States. And then since then, how it's being maintained at a like national, regional, and hopefully eventually local level. So I have West Nile virus is a big interest in mine. I have been trying to get the NIH to fund a US-wide genomic surveillance program for West Nile virus. That's been hard to get set up, but hopefully we eventually get this. But this is a sort of approach that I'm going to use throughout the rest of my talk. And it's really, again, to get focused on these types of questions. Is how do virus genomics help us to answer questions about disease ecology? Because if we understand how they emerge and how they spread, we can start to think about what are the landscapes, what are the vectors and hosts that are involved in creating the patterns that we can recreate with genomics. I'm going to give this talk through 3 vignettes of 3 of my current favorite viruses. Powassan virus that is maintained by ticks and small mammals. Eastern equine encephalitis virus is maintained by mosquitoes and birds. And then Jamestown Canyon virus by multiple different mosquitoes and large mammals. I like these examples because they're quite distinct. And what you'll find out through this is how their patterns are actually quite a bit different from one another, even though they're all arboviruses. What's also fun is each one of these stories takes place right here, right in New England. Massachusetts is a focal point in these studies. And so I like to, even though I do things where I talk or work globally, I really like to have some projects that are right in my backyard as well. So the basis of all of these viruses that usually don't get a lot of attention is that we need to set up systems that we can sequence them all. So in my lab, I talked about how we sequenced 25,000 SARS-CoV-2 genomes. That's not to make you impressed, though you should be impressed. But it's to say that we have this like machinery, this mechanism to bring samples in and process them quickly. Instead of setting up a brand new sequencing pipeline for every new virus that we have come in, what we have are some very standardized approaches in which now we take what is the COVID-seq protocol, and all we do is, in one part of it, is we flip out the primers that we use, make a little bit of modifications in bioinformatic protocol, and it's the same protocol that is used to sequence everything. And so in some ways, my technicians kind of hate this because they get a little bit bored because they basically do the exact same thing all the time. Doesn't matter what the virus is. Sometimes we just have to do a little bit of upfront R&D to get the conditions right for them. But this even works. I'm actually like, I feel weird saying this as a virologist. Like I'm starting to work in bacteria, which is kind of gross. But we're even doing, we have this set up to do Mycobacterium tuberculosis sequencing, TB, which is like 4.4 megabases. And with 5,296 primers, we can sequence the whole genome in 2 reactions using the same thing that we do here for 888 and Flossin and SARS-CoV-2. So these protocols we put out into the world, other people are able to quickly adapt them. So there's more and more capacity for sequencing. So we can use this technology here for viruses that need more love. The other thing is partnerships. And it's one of the reasons why I'm here. I know Steve because I reached out to him to say, hey, can you help me? And I do that with a lot of different people. And mostly I find that people are pretty excited to collaborate. And so this is a network of, I'm going to say public health partners, but this could be mosquito surveillance programs. It could be academic universities. It could be state labs that do arbovirus surveillance, mostly for West Nile virus is the base of it. And with these partnerships, we can reach out to them in different projects that we're trying to get involved in to say, hey, if you have some of these samples, can we do this sequencing with you? We'll give you data back. We'll work with you on what we'll end up doing ultimately with the data. But for me, like I can't be everywhere trying to do surveillance. Like this is a very hard thing to set up. Like there's really good programs for it. People do a lot of work. What we're trying to do is help give people more data for the work that they're already doing. So even on here is UMass in this location. But most of the work that I'm going to present here today are from two main partners that we have. One of them is the Connecticut Agricultural Experiment Station, which is literally in my backyard. I look out my bathroom window and I see Phil Armstrong's office there. And they do mosquito and tick-borne surveillance from throughout the state. They've been doing it for more than 20 years using the exact same trapping locations, which has been fantastic to have consistent data. And then through the Wadsworth Center in New York, they also have very robust surveillance programs. And I'd love to be able to add more places on here, but most of what I'm going to talk about now is primarily through these collaborations. So get into the first vignette. I'm going to talk about Powassan virus. Just to point out that this is a story that we had recently published. As Steve said, that this is led by, it was a project led by Chantal Vogels, who is now, was a postdoc with me and is now an independent faculty member at Yale. And she is now taking on this program to run with it because, as you'll see from this, we unlocked a lot of really new and interesting questions with Powassan. Okay, so Powassan virus was a really fun virus to get to know because with something that is tick-borne, it has a much more complicated life cycle than the sort of the rapid pace of things that I know of through mosquito-borne viruses. One, and you'll probably know this very well, so I'll try not to over-explain, but they have long lifespans that go through multiple different seasons, different stages of the ticks will feed on hosts. There's two main lineages of Powassan virus. There's this lineage 1, which is thought to be maintained in more of this not as much urban human interaction environment with Ixodes cookii, which is a nidicolous tick that lives with its hosts and a bunch of small mammals. Genetically, these viruses are very distinct from the other lineage 2 viruses. These are not as known to be causing human cases. In fact, hadn't been detected for since like the 70s until we did some sequencing studies that actually found that it does exist. And it might actually exist in Ixodes scapularis as well. I don't think that the systems that I'm putting up here are all that cut and dry. Lineage 2 is what we're more concerned about because it's primarily vectored by Ixodes scapularis, which is also vectors of Borrelia and Babesia that cause a lot of human disease. We know that the different stages of them will feed on humans, which is where the risk here occurs. This may not be exclusive to Ixodes scapularis. We found it in actually Dermacenter as well. And it's maintained with small mammals. I have a white-footed mouse up here. That's kind of like what the dogma of it was, but I don't know if that's actually really true because we don't know a lot about actually how this is maintained. And then also in here, I just want to point out is that there's two main foci of lineage 2. There's here in the Northeast and in the Midwest, and they are very genetically distinct, though we did just find a Midwest lineage virus here in the Northeast, so it's not, again, clear. But the point is, we have some diversity. There might be some differences in the ecology of these viruses, but also, as you're probably already guessing, there's a lot that we don't know. But the important part of it is that human cases of Powassan are increasing. And we certainly are not catching the majority of cases. Most of the cases that we know about cause severe disease. This has something like a 30% case fatality rate. It can cause encephalitis. But certainly a lot more people are actually being exposed. It's hard to know if these increasing numbers are just due to increased case ascertainment, awareness of it, or if it's actually something to do with more transmission or more human exposure or some combination of all of those things. But when you have very severe disease, even a few cases are important. So going into this, we had a few basic questions. Focusing on lineage 2, which is thought to be most of these cases, We want to know, when did this actually emerge into the Northeast? How fast does it spread between places? And how is the virus being maintained? Pretty basic questions, but some that we can actually answer with phylogenetics. Starting this project in 2018, we go to our friend GenBank and look to see what sequences are available. And at that time, there were less than 40 complete Powassan virus genomes available, not enough for us to actually do a whole lot with. So we got to start reaching out to friends and colleagues, collecting samples and sequencing viruses. In the end, from this study, we sequenced over 400, which doesn't seem all that impressive when there's 17 million SARS-CoV-2 genomes that have been sequenced. But for something like Powassan, this actually provided enough data where we can start to investigate. Okay, so here's the first result. Let me just take a drink really quick. This, again, like I was showing with West Nile virus, is a time-resolved phylogenetic tree in which we are trying to reconstruct the location of the ancestors in time. So the locations, the colors show location. Green is New York, the purplish color is Connecticut, blue is Massachusetts, and then there's some red in here that is Maine. One of the first things that we got from this is that the time of the most recent ancestor of all of the sequences is in the 1950s. With an confidence interval between the 1940s and 1970s. And so this at least made sense to us in some ways. And what the thought process here is, just like with Lyme disease that probably came in much earlier than this, but we had this process of the forests in New England coming back. With that, you had the white-tailed deer coming back, and here is a deer population size for Connecticut and New York, and you can see how much they've started to grow. And with the deer came the Ixodes scapularis ticks. And so the thought had always been that Borrelia comes in a little bit before Babesia, which comes in a little bit before Powassan, and I think that still makes sense here. Powassan could have been in the Northeast before this, and then was eliminated when most of the deer population was gone, and then reemerged. So this might not be the first time, but many of y'all are probably from this area. Why were the forests so depleted, and why did they come back? Yeah.
The farm,
The shipbuilding, certainly one of them.
the farmers got tired of digging up giant rocks.
Yeah, the farming. I think, honestly, the farming is the real answer on this. Like when farming really went to the Midwest, right? Like when y'all like take hikes through the woods, you can see like the stone walls that are still exist there that represented where the boundaries of fields used to be. And now we have all these young forests. We were talking about this on the way in. These young forests that exist now in these places. And so we actually had, not as many forests back here in the 1800s, so therefore not as many white-tailed deer. The thing that I like to think about with this is the reason why that I know is farming is the real answer, but I think more about like Moby Dick and like the shipbuilding for the whaling industry and how that when that went down, then there was not as much of a need to build those ships. Maybe that had a small piece to do with it, but I think farming was probably the main one. But the point of it is that we had this net positive effect on our overall ecology here by bringing forests in and bringing in white-tailed deer, but there's also some things that come with it, like bringing in ticks and then therefore their pathogens. Okay, so we know that it probably came into the Northeast around the 1950s, and then what we see when we actually do the phylogeography of the entire area is that we have this mostly south to north spread of the virus. And what we also had found from this is that there's very slow local spread, followed by several long distance introductions. And so the long distance introductions we think are actually due to birds, eventually like carrying infected ticks to new locations and they start new foci. But for most of it, we see it's very geographically limited, but the virus does move. We estimate that it, from this either southern New York, southern Connecticut, when it came into this region, like I said, in the 1950s, by the early 1990s, it finally made its way to Maine. It might still be moving north. It still might be moving west as well, and maybe even a bit southward. But there's still probably an expansion here that we haven't quite captured the whole thing on this yet. But when we put this into, when we have time and location, we can estimate dispersal velocities of viruses. And this is always one of my favorite things to do, is like, how fast is something spreading, right? And what limits or drives that spread, and the context of all of this. So Powassan virus spreads at a rate of about 3 kilometers per year. And remember, again, this is like, there's a lot of over dispersion in this, probably very, very little spread, and then boom, a big jump by a bird. But an average is about 3 kilometers per year. This is very similar to Lassa virus in West Africa that is mainly maintained by rats. Things like rivers are a barrier to Lassa virus movement because the rats don't move over that. Similar to rabies virus in skunks, when you put rabies virus in an animal that has wings, they move a little bit faster. And here we have avian influenza, West Nile, and yellow fever virus, and they move quite a bit faster, to the point that, oh, I didn't have this up here, but I think it's like West Nile virus, when it emerged and spread throughout the United States, spread at a rate about 50 times faster than that of Powassan virus. Another way to really make this point, so this is just a different version of that same phylogeography. This is called continuous phylogeography, where we're kind of mapping how it evolved through space without giving it restrictions. And when we look at Connecticut here, we had, there's sampling sites for ticks all through here. In between all these sites, all the ticks are negative for some 10, 20 years that they've been doing this. They're all negative. It's these sites that continuously have been positive. But if we look at 2 of these sites right here, Reading and Bridgeport, about 12 miles apart, we know that adult ticks can feed on deer, and we know that deer can move. Now, some of them might stay close to a source and not move miles every day, but certainly the bucks, when they're en route, will move. So the deer can move between these locations. The thing is, these viruses in these locations, without having any positives in between, have been separated for 33 years. And this is one of the, I think for us, one of the more important insights here, and how strictly tight these foci of transmission are, that helped us develop some new hypotheses about how Powassan is maintained. First, that we think that vertical transmission is probably really rare. So what I mean by that is that we think that the adult ticks, once they're infected, they're mostly dead ends to transmission. So even though they're getting, the ticks are getting on, adult ticks are getting on deer, and they might be moving around, if they were then transmitting that virus to their offspring, which was thought to be very common for Powassan, you would actually see more geographic distribution of the viruses because they're following the patterns of the deer. But rather, they seem to be more following the pattern of small mammals that actually were rivers and roads could be boundaries. So we actually think that the majority of the transmission happens right on the back of, in this case, a mouse, where you have the nymphs that can pass the virus back down to a younger generation, to the larvae, either through co-feeding transmission. So as one is feeding, another one's feeding next to it, the virus is getting directly in, or through the mouse actually getting viremic, because these stages here can actually occur at the same time. So this is something that Chantal is investigating, the size of these foci, more about these patterns here. But I think the cool part about it, since you do have such tight foci for transmission, is if you can identify them, you can go in and you can remove the ticks from that area and effectively remove the virus. It's not just like coming back in the next year. You might actually have long-term protection against Powassan in that case. All right, so we're going to make a jarring transition to Tripoli. We're going to do this transition one more time. You got a question now?
Can I ask a question before you go away from Powassan?
Yeah.
So at least some thinking is that before the pre-Columbian density of white-tailed deer might be as high as it is now. There's some thinking that maybe Native Americans managed the habitats in a way they did control burns and so. maybe some of that distribution reflects more of a relic model instead of a U distribution. Would your models be able to detect that as a parsimonious explanation of?
I don't think anything that was happening before the 50s, we wouldn't be able to detect because none of those viruses, none of those lineages from back then had persisted till today.
I guess what I'm asking are those networks constrained by some route that wouldn't allow for another origin and another origin? At some point you start to say, well, it's easier to explain, not that this made it from here to there, but that was there originally and now is spreading from that point.
Likely not because of the time to the most recent common ancestor. They all shared an ancestor in the early 1900s. So there could be a single relic that came from there that hit out here someplace in probably southern New England. That could be the relic part of it. I don't know where it came from before the time of this tree.
Right.
It looks like it's south to north, but was it hanging out in the south or was it actually just hanging out in New England? Phil and I have talked about this a lot, and it would be really interesting to be able to do. Yeah.
Do you have any ideas about sort of the transmission of the human disease, the severe human disease? How does that match with sort of the movement of the virus that you've been tracking through this phylogeography?
So the humans would be dead ends for transmission. And we have very few human genomes from humans because by the time you come in with encephalitis, the virus is gone and we can't sequence it often. Sometimes you can get it if someone dies, you can get it from brain tissue. But if someone is alive, that's a little harder to do. But the idea is that we actually had more human surveillance. What you could do is if you sequence the genome from somebody, right, and based on its relatedness with everything else we have, we can actually figure out maybe where they got exposed because the virus is not moving that much.
And so was there a big jump to go from lineage 1 that doesn't seem to be associated with severe human disease to lineage 2?
I think it's purely ecological. I think it's just because lineage 1 is mainly in ticks that don't feed on humans. Okay, Tripoli. Now we're on to mosquitoes. This work was led by a postdoc in the group, Verity Hill, and an MPH student, Toby Koch, who is now a PhD student with Greg Ebel, to feed the inbreeding in this field. Okay, so Tripoli, y'all probably know a lot about this because are we in the Tripoli area here? I have to put myself on the map.
Belchertown has a bunch of.
Okay. Tripoli is a swamp virus or a wooded wetland virus, if you will. It's maintained by a lot of different passerine birds and mosquitoes, mainly Culicidae melanura, and it can cause severe disease in humans and horses, hence its name, where these are dead ends for transmission. One of the things that I was working on right before the pandemic, and I had to stop to go do other things for a bit and then come back to it, is in 2019, we had the biggest Tripoli outbreak on record, right? In both cases in humans and in horses. I remember this distinctly of all the news about should we spray, should we not spray, should kids be having soccer practice and baseball practice in the evenings when mosquitoes are out. It was now seems like a drop in the bucket in terms of hysteria around public health. But at the time, this was okay, a big deal. And so, in Connecticut, there were only 4 cases that were known. All of them treated by our hospital. One of my close colleagues was the attending on all of them. 3 of the individuals died in the hospital. One of them had long-term sequelae and just passed away last year. Right? This has a very high case fatality rate. There is a vaccine for horses, but unvaccinated horses often suffer the same fate as well. So even a few cases are really important here. And this just to show that most of the cases themselves happen in the late summer, early fall. So 2019, this was happening. You start to see a little bit of patterns in here of ups and downs and stuff. But we came into this project trying to understand okay, so how is this virus maintained? Again, similar to the Poisson thing. And then also, why was 2019 so bad? So we started with sequencing. Triple E is a select agent. The RNA of triple E is a select agent. This makes it a bugger to work with. It means that a lot of surveillance programs, the moment they detect triple E, if they do not have select agent status, they immediately have to destroy their samples. This is not an easy project to go into. My lab cannot work with this. So I worked with Alex Siota's group at Wadsworth, and they did the sequencing for this project. They sequenced 80 new isolates, 48 from 2019, a bunch that were from Connecticut since the first time ever, but from our region of states of New York, Massachusetts, and Connecticut. Most of them coming from Culicida melanura pools that we think is the primary vector, some from Cochleatidia perturbans, which might be a bridge vector, and then a few other sources here. And I always love the random turkey in this. And you can see that here's the distribution of where our sequences come from. And then we pulled this with other data. So you can see that triple E's in the Northeast, but it's also in the South. So we started with a phylogenetic tree. You all know the drill now. This is a time resolved discrete phylogenetic analysis. Because I did this work after the pandemic, the first thing we had to say is, no, this was not caused by a new variant. Right? This was actually caused by several different lineages of the virus that we found here in 2019. So the virus is not to explain. Rule that out, go on to the next. When we looked at this phylogenetic tree here and looked at all the movements, we noticed something that was really interesting and really cool, is that the backbone of this tree is mostly blue. And meaning that the ancestors for all these viruses are somewhere in Florida. And when we map this out, it looks like, gosh, there's a lot of spread from Florida to the Northeast. So we simplified this a bit, and we took the tree and just said, Northeast or not Northeast. And we found all of the clades that were in the Northeast. And then we split those out and just plotted them in a way where the first dot represents the introduction time inferred from the phylogenetic tree. The second dot represents the first time we sequenced one of the cases in that cluster. And the last dot is the last date of a sequence from that cluster. And we just plotted them over time, right? And here's a histogram of it. The point is, what we see is these are all independent introductions into the Northeast, and they don't survive here very long, only about a few years. And so the triple E in the Northeast is dependent on new introductions. Again, very different from the Boston story where you just like, it is here, it's going to be here for a long time and it's not going to move. Triple E doesn't last here. It's not fully endemic to the Northeast. Without new introductions coming in, we wouldn't have it. And the source is really with my colleagues from the Florida Department of Health. It seems to be the panhandle of Florida where you have year-round transmission and maintenance of the virus that happens. So if you have this need for new introductions constantly happening, what led to the large outbreak in 2019? I'm going to save you all a lot of time by the things we dug into and found that probably are not the case. This was not expanded range. Everything we found in 2019 fit within the standard footprint that we've known of triple E before that. It was not case demographics. The people that were infected in 2019 are very similar to the distribution of people that are infected before. Human behavior, this is kind of wishy-washy on how we did this. We looked at cell phone data and outdoorness, how much time you spend outdoors to see if 2019 there was like some crazy like Woodstock party that everyone was at or something, but people weren't outdoors anymore in 2019 than they had been in previous years. That's probably not it. We tried to look at a bunch of factors with bird abundance. Bird abundance and virus transmission is kind of a complicated thing. And there's a lot of different birds that are involved. We could not find any clear relationships with bird abundance in 2019 that led to this. But what we couldn't measure is bird immunity. Certainly bird immunity would be a part of this, right? It's kind of a missing piece of this equation. But what we were left with, it was a lot of mosquito surveillance data to go and look at. So this is New York, Massachusetts, and Connecticut. This is from the Mass DPH. The bars are infection rates, and the lines are the number of Culicida melanura tested. One of the things New York actually didn't really have much of an outbreak in 2019. There's a few cases, but we really saw a lot of the increased action was in Massachusetts and Connecticut. You can see here, 2019, huge infection rate, a lot of mosquitoes detected compared to previous years. When you plot this here, you see a very positive relationship between the number of positive Culicidum melanura pools and the number of human and horse cases. That makes sense. When we look at Massachusetts and Connecticut data further, look at the Culicidum melanura abundance, 2019 was pretty high. The infection rate for triple E was fairly high in those years. The vector index, which is the multiplication of the infection rate by the abundance, showed that this was relatively high and fairly early in the season. And index P we'll just kind of ignore for the moment. So there seemed to be something that was happening here with the mosquitoes. There's a lot of infected mosquitoes that year. So we had these data, we're presenting them back to the Mass DPH and to, yeah, go ahead.
Sorry, I didn't mean to interrupt you, but I'm noticing Rhode Island is not there.
I didn't have Rhode Island data for,
Okay, because
Yes,
I
study.
live in Bristol County, right on the border of Rhode Island and Mass, and so like during the 2019, there were cases of West Nile in it, and that directly affected our like public health like impacts and the restrictions and things like that. So I was wondering, like, did you just not have access to the data, or was it not collected?
for Rhode Island?
Yeah.
Yeah, because we didn't have any sequences from there, and we had this as like as a genomic-led study. I think they're included in that first figure where I'm showing the general Northeast, but we didn't have any, at that point, we didn't have access to anything to really include. And we also didn't have, at that time, the access to all the mosquito data for this. But I'd imagine the story in Rhode Island is very similar to what was happening in Massachusetts and Connecticut.
Thank you. Sorry.
Okay. So we're having these conversations with some of the folks that are working in the public health systems here. And they ask, okay, with this, with this information, can you create a model to predict outbreaks? Right? That's always what we want to have. And my answer was, well, no, we can't do that because we're depending on these introductions from Florida. Right? So it's a really hard thing to model what is happening in two different places. Though I think with more information about Florida and information about, and I forgot to say this, but hopefully y'all were putting this together. This is probably bird migration is bringing it from Florida to the Northeast and dropping it off here, right? If we had better understanding of all of those dynamics, maybe we could put together a prediction model that also include local data about what makes for a big mosquito year, right? And bird immunity, all those things. It's probably possible, but at the time that we're doing this, it wasn't possible to set up. But I said, we can use mosquito surveillance data where we might be able to create a risk map for that year. Because one of the things that we noticed from 2019 is not just more mosquitoes and higher infection rates, but it was like a little bit earlier. Yeah.
So all of the data that you've been collecting are from single mosquitoes, or do you pool data?
Cool. These are pooled surveillance programs usually run some pooled mosquitoes. So our infection rates are maximum likelihood infection rates based on that. So our MPH student, Toby, put together this statistical model where we had a bunch of covariates. And what we were trying to do is predict cases using our covariates based on actual number of cases that happen so we can understand the important factors of different things. The main thing that I wanted to point out here that we found from this is finding a positive mosquito pool. Not the numbers of them, but just yes or no. Do you have a positive mosquito pool in June or July? A 220% increase in the chance of having human cases later that year. But early season transmission was really important. So yes, we can't predict out years in advance right now, but I'd love to be able to do, right? But you can use that early data to say, hey, this is going to be a bad triple E year. And I think, what is this? Is this 2025? Yeah. 2024, there was some triple E activity. And so we were able to put that to play where we actually found early season positives, but we actually didn't have a whole lot of cases that went with it. So I don't know if that's intervention wise, sort of luck, because this is a few number of cases overall, or just our model is actually crap. So to end the triple E story, and I'll try to get through the last one quickly, is that outbreaks in the Northeast are self-limiting. The virus lineages likely go extinct within a few years due to a buildup of herd immunity and an RNA that probably doesn't go above 1. Also, I bet transovarial transmission like is important for the maintenance of West Nile virus and stuff is probably not super efficient in the Northeast. It probably says something in Florida that you have a lot of year-round transmission. So you just need like really brute force of a lot of infected mosquitoes to keep them on the next year. And once you build up some herd immunity, it's probably enough to crash the system, and it needs to be reintroduced when all the conditions are right again. And again, the potential for outbreaks needs new virus introductions, and that currently none of this is predictable beyond a couple of months, unless we knew more about the source.
Is there any correlation between new virus introductions and severe hurricane seasons in Florida?
These are some of the big things that I haven't looked at those data yet because hurricanes are really complicated with all this because initially they wash away mosquitoes and eggs and such. And then afterwards you leave a lot of standing water for breeding. I know that this is an impact on like dengue and Zika in Florida, right? In like Miami. But I don't know how that impacts the panhandle at this moment, but that's overall weather conditions, El Nino events and stuff probably are part of the drivers here that we need to put together. Yeah.
What's the standard for number of mosquitoes per pool? And also for your model, do you have a number of pools that you try to test per site?
So the max is usually, this is not the same for every system, but for the programs that I know, the max is usually 50 per species per trap per collection would be the max you would use. And sorry, what was in the number of pools? It would be everything from that trap and how many pools that makes gets tested unless there's some economic resources for testing. But for both of the programs I showed here for the Mass DPH and for the Connecticut Ag Station, I think every pool was tested. At least they had for Chelicida melanura. I think that's not right.
I don't know if they do every pool from each trap.
No, like in Massachusetts, I work for Massachusetts. It's 50 mosquitoes per pool. And then because of budgetary reasons,
Yeah, maybe
depending
there's a
on like the
max number.
district, we have caps, but
You have to spread it out between the different districts.
Yeah, there's probably a max versus a min. Okay. I quickly want to move on to Jamestown Canyon. These are preliminary data. So the last two things were published stuff that we're trying to push on. This is something new. This was started by an MPH student that graduated last year. Basically, she had access to the Connecticut Ag Station surveillance data and said, hey, I've never really done much with Jamestown Canyon. Can I jump into this? Oh, please. So this is a really, really fun virus in that this is how I think it's maintained. And this is way too simplistic, I'm sure. First of all, there's a lot of mosquitoes that you'll find positive for it. From Connecticut, there's at least 26 species that have been found to be positive for Jamestown Canyon virus. Some you will find from a lot of sites and a lot of number of years and have a lot of isolations like Aedes canadensis. Then you have a bunch of minor species here that who knows if they're really playing a role. But the point is there's a lot of different mosquitoes involved in this. And the amplifying host is thought to be white-tailed warbler or other large. Again, I don't know exactly how true this is, but this is not a bird system like with Tripoli. This is the second most common arbovirus in the United States behind West Nile virus. It's found in many different states. These are just the human cases. 13 have been recorded from Massachusetts, 2 from Connecticut. Increasing numbers in recent years, again, probably due to case ascertainment. This is important again because it can cause encephalitis and other severe disease when we don't know or we have underdiagnosis for most encephalitis meningitis cases. Arboviruses could be an important cause. We don't know how much Jamestown Canyon is a part of this greater picture of things because it's not something on clinicians' mind, right? There's not clinical test for it that people are using to look for, like, I have this encephalitis case that's really severe. Is it Jamestown candidate, right? In a lot of cases, they just want to know, is it bacterial? Yes or no? Because if it's a bacteria, you can treat it. If it's a virus, I can't treat it anyway. So why am I looking further? So we don't really know the true clinical impact of it, but it's an interesting virus because it's so widespread. It's also interesting and a pain in the butt because it's segmented, right? It makes sequencing a little bit harder. It means that they can reassort as well. Another cousin of this virus, or Boucher, is emerged from Brazil and is now spreading through different parts in the Americas. We had a lot of travel cases in Florida, and that is thought to be due to a novel reassortment of the virus. So starting this project, we went into a few different things here. Similar questions that I've mentioned before is what mosquitoes are important for virus persistence. Like, can we disentangle this web of all these different mosquitoes and figure out what we need to focus on here? When was this virus introduced? How is it spreading? How often does reassortment occur? For the sake of this talk, I'm not going to go into these data here. I can say it's very little. We estimate about 1% of the time that these reassort. Okay, so to start this project, we had 20 sequences available. And then Ellie went in and sequenced almost 700 as a part of her MPH project. Some of them that came from here. We have a few, hard to see into this. There's a few from Massachusetts. Most of these are from Connecticut because we had this right down the road. Alex gave us some from New York. We have some from Pennsylvania, North Dakota, and Michigan. We're trying to expand this out more, but we at least have a good basis to start with here. And then this is, for the most of the rest of this talk, I'm just going to talk about Connecticut for now because these are the data that we have. And for those of you who don't know what voltine means here, I'm explaining we're starting to split these mosquitoes up into two main systems. We have these univoltine, or these single brood mosquitoes. And these are mosquitoes that hatch in these big bursts. And when they hatch, they go and take blood meals and lay eggs and take blood meals and lay eggs. And then when those mosquitoes die, right, you're not getting another generation. Those eggs will not hatch again until the following spring. So you get one cluster of them. Multivoltine mosquitoes, these are the things that are transmitting West Nile virus and Triple E. They have multiple broods per year, and then they'll overwinter as diaposing adults. And so when you look at the abundance of them, I know you can't read the species names here, but that's okay for this moment. Most of these are Aedes mosquitoes. You see this big peak in abundance versus like this broader spread. We can look at this in a different way. Here's the univoltine mosquitoes. Here's the multivoltine mosquitoes, and their mean abundance per year. Here's now their infection rates with JCV. You can see a higher infection rate in these univoltine, a little bit less than the multivoltine. And then when you multiply these together to get the vector index, you see this really high vector index in the early part of the summer in June and July in the univoltine mosquitoes. So this is some preliminary analysis that I just put together this slide yesterday that Ellie was doing. This is very similar to the phylogeographic analysis that I was showing you. But instead of location, what we're doing is using class of mosquito, multivoltine versus univoltine. What we're trying to do is estimate for ancestors. Did the ancestor of these viruses, did it exist in a univoltine or multivoltine mosquito? And what you can see throughout here is all this blue is that the genetic diversity of the virus is maintained in univoltine mosquitoes, and the lineages that are in the multivoltine don't last for very long. This is a divergence tree, not a time tree. We need to scale this over to know how long they might last in these systems. But this gives us a hypothesis for how the virus is maintained. We think JCV is maintained primarily in these univoltine mosquitoes. So they emerge infected. They go and feed on deer. Deer become viremic, infect other mosquitoes that feed on them, including other multi or univoltine mosquitoes. Those mosquitoes lay their eggs through transalveolar transmission. Some of those eggs are already infected, so when they emerge the next year, JCV, right? this actually gets into an interesting thing where maybe this virus is actually evolving to become insect specific, right? Where it doesn't even need to have the deer here. But I still think the deer is probably important for amplification and getting more lineages going. Then from the deer, these can spill over into the multivolting mosquitoes that then can have the sort of risk for JCV extend throughout the season. These may be the most important ultimately for human infections, as you have a longer time period in which you could have infected mosquitoes that are out there. But ultimately, we think that whether this is within a year or just a few years, that these are dead ends for the virus, that it really, without the univolting mosquitoes here, these would end. Next, we need to split this up into say, which ones? Because there's a lot of different Aedes and anomaly species that this could actually encompass. We think that there might be a couple that are important here. That we could actually understand like what mosquito species are really driving this from a year-on basis. This would help with creating predictive models, right? And then understanding what mosquitoes then are important for human risk later that would also help with those predictive models. Kind of two systems on top of each other. Hypothesis. The other thing when we're sequencing this is that we found that there are two major lineages, A and B of JCV. And you can see, I think we got 6 mosquitoes pools from y'all that we sequenced. They're split down the middle of 3 and 3. And Connecticut, this is mostly lineage A, about a quarter of them are lineage B. The reason why that doesn't look like there's any lineage B in New York is because when we were doing this work with them, we figured out that their PCR primers for detecting JCV misses lineage B. So they need to update their primers to capture all of the JCV. So, and here's the temporal analysis. This is just through Connecticut, right? That we see both A and B exist over the last 20 years together. A is just at a higher rate. And here, now this is looking at every single trap that we have in Connecticut in their distribution of A and B. And what was interesting here is that you have the Connecticut River going through here. Is this, you have mostly B on this side and mostly A on this side, which got us to thinking, is there some ecological factors, right? Different species that are maintaining them, different times of rates of invasion, other ecological factors. So here's some other really preliminary analysis. Ellie was doing some continuous phylogeographic analysis and was plotting these. And so I started putting them together. And so B is in blue, lineage A is in red. This goes back to 1938. By this time, we had at least detected 2 separate introductions of JCV into Connecticut. This one, B, had already spread a bit. I need to get that exact introduction time, but probably early 1900s versus A was just getting in here. Fast forward 10 years, you can see it move a little bit. Fast forward 10 years, 10 years. We get to 2023 at the end of the study time. One of the things, and we have to calculate this rate, is this is from 1938 to 2023, a lot of years here. It actually didn't move that far. The dispersal velocity is pretty slow, right? I don't think quite as slow as Powassan virus, but we need to look at that. But certainly not the speeds of what we see of spread for West Nile virus. So I think this is pretty clear that birds are not involved here, but we still need to understand the patterns of thread. But I think mostly what we see is the distribution now of A and B at the different traps are just the fact that they were introduced into 2 different spots, one side of the river each, and because they just don't spread all that fast, you're still seeing the differences, but maybe you fast forward this another 50 years, they might be equally distributed. But we're still going to look at some of the ecological factors. I don't know if they spread at the same speeds yet, or if there's other sorts of things there.
What's preventing spread to the west, or did you just not sample past that?
This is only using data from Connecticut, so that's the reason why. Because the sampling kind of around these sites is a little bit more patchy. So some of these might be doing this instead of like this, and certainly going out and radiating. So preliminary findings here is that we think that these univolting mosquitoes support persistence while others may contribute to human risk. at least 2 separate introductions into Connecticut more than 70 years ago. Very slow spread, and we're interested in what is sort of like putting the cap on that speed. And that this intralineage reassortment, like I said, I didn't show you, but it's quite rare. So I hope that I've showed you how genomics can help us uncover patterns of emergence and spread, help us understand how the virus is maintained, maybe even points of intervention once we really understand this maintenance process, and explain why we should be doing more of this work. Because all of these projects involve little sequencing data. We expand that a bit, and we learn a whole lot more. It's kind of like low-hanging fruit in some ways, and it provides a lot of new hypotheses for us to investigate in the future. A lot of different people on all of these projects to think. Really thank you guys as well for being early partners on some of this work. Hopefully we can expand that. And also just wanted to say, if you go to my website, grubauglab.com, I have a little tab here called Nextray, and all of our data sets are up there if you want to play with some of these yourselves. And here's my contact information.